Okay, so these two questions really launch our discussion of meiosis. Do parents give their whole DNA copy, so their whole genome, to each offspring? Of course, the answer to that is no. The chromosome number would double each generation if it did. So our chromosome number is essentially constant for a species. So we say humans have 46 numbers. These are diploid numbers of chromosomes. So that's a 2n equals 46. Corn has 20, onions have 16, dogs have 72. If you have more than 72, or if you have 71 or 73, you're not a dog. If you have 44 or 41, you're not a human unless you've got some sort of a disorder that throws off your number of chromosomes. Here we see our sexual life cycle if we only performed mitosis. So let's start down here where humans, there's a human male, a human female, goes through mitosis, makes a sperm cell that has 46 chromosomes because mitosis only makes clones. Those two gametes fuse to form a zygote that has 92, which is now no longer human. So our chromosome number would double every generation, so we must need a method to reduce that number. So we go through this process called meiosis, where we take our 46, cut it down to 23. Females do the same thing, donate to the zygote. We get the correct number of chromosomes in our offspring. I use that term gametes. Meiosis is used to produce these gametes, which are sex cells or spores. The purpose of meiosis is to reduce the chromosome number by half, which in effect prevents the chromosomal doubling each round of reproduction. So a sexual life cycle is going to have alternations of meiosis and fertilization and mitosis to keep chromosome numbers constant. Here's what we're talking about. In a human, we go through meiosis, we create gametes, gametes fertilize, then we do a whole bunch of mitosis, and the next generation continues the sexual life cycle. Goes through meiosis, fertilization, whole bunch of mitosis, then eventually meiosis, and so on and so on. Another vocab term here, ploidy, is going to be the number of chromosomes in a set. So N is haploid. That's our number of chromosomes per species. So in humans, our haploid number is 23. Okay, so diploid is going to be two sets of chromosomes. Di, of course, for two, and ploid from ploidy. So that's our most common number of chromosomes in our body, or our somatic cells. Our diploid number, of course, is 46. Corn's diploid number is 20. Notice that 2n number there. Fruit flies are 8, which is really important when we get to learning about Drosophila and all the genetics that come from that, but we'll talk about that later. So human chromosomes, we know a little bit about karyotypes because we've done a lab on them already. Human somatic cells, so all of our body cells that aren't directly involved in making babies, have 23 pairs of chromosomes, so our diploid number is 46. The two chromosomes in each pair are called homologous chromosomes, or homologs. Chromosomes in a homologous pair are the same and carry genes controlling the same inherited characteristics. They're the same length and carry not exactly the same genes. One came from mom, one came from dad, but they're very similar. Here you see a rough cutout of an old school karyotype. This is an old picture that we used to get. We would wait until the cell was at a certain phase of mitosis, which you should know, when the chromosomes become visible. Then we would basically just freeze frame it, and then we'd have to cut them all out and arrange them and put them in order so that they looked all nice and neat like this. We used our length of our chromosomes and our banding patterns to figure out what matched with its pair. Notice this is a human female. We can tell by the two X chromosomes in the bottom right. Again, we're going to use our length and our banding patterns to figure out what goes with what. This we've got a color printout that's definitely much more modern, more representative of what you'll see in a lab today. Certainly you'll still see some black and white printouts, but you see we've got a male here because we've got an X and a Y. Notice how much shorter the Y is. That's again going to be really big later. And then just kind of a review of those terms. So each pair of homologous chromosomes includes one chromosome from each parent. 2 sets of 23, one from mom, one from dad. We know all that kind of fun stuff. Diploid cell has 2 sets of chromosomes, so we should be good there. Haploid set, think half set. When you see haploid, it's going to be a single set of chromosomes. So that for humans, that's going to be 23. For corn, it's 10. For fruit flies, it's 4. Those are going to be the number of chromosomes found in the gametes or the sex cells. So gametes are haploid, so they get this N notation. Our haploid number is 23. Each set of 23 is 22 autosomes and a single sex chromosome. Kind of some more vocab there. In an unfertilized egg, the sex chromosome is X. In the sperm, the sex chromosome may be either X or Y. That's why lots of people say the dad determines the gender. Polyploids are going to be entire sets of chromosomes. So we're not talking about a trisomy here. We're talking about a polyploid. So we've got an entire extra set. So instead of being diploid or haploid, now we're tetraploid or we're triploid. Really common in plants, but most of the time in animals, it's fatal. So here are some variations of the sexual life cycle we see in animals. This is the one we're familiar with. Some plants and algae create these things called spores that may be around for thousands of years. They're essentially eggs that can last a really, really, really long time. And then some fungi and other protists have a slightly different version where this haploid unicellular or multicellular organism comes in to fertilize, gets some external fertilization. Plants and algae exhibit alternation of generations, so that's going to involve the spores and the sporophytes that I was talking about earlier. It's going to the life cycle as a whole is going to include both a diploid and haploid multicellular stage. So that's what we're seeing right here, and I'll provide some more information on that in a subsequent lecture. Okay, so in plants, each spore is going to grow by mitosis into a haploid organism called a gametophyte. The gametophyte is going to make gametes by mitosis, and then the fertilization of the gametes results in a diploid sporophyte. So that's essentially this alternation of generations that we're talking about. Like I said, I'll post some more information on that because I know I'm not doing a good job of explaining it. I'm not feeling well today, so I'm kind of stumbling through my words. But another variation, I talked through this one on the picture. That's that far right picture. The key is that there's no multicellular diploid stage. The zygote produces haploid cells through meiosis. During that, each haploid cell is going to grow by mitosis into a haploid multicellular organism. So there's no two sets. There's only one set of each chromosome. So differences of meiosis and mitosis. 2 cell divisions, not 1. We're going to produce 4 cells, not 2. The synapsis and chiasmata are going to be observed in meiosis. That's essentially crossing over, if you remember that term from Biology 1. Okay, 3 events are unique in meiosis, and they all are in meiosis 1. So synapsis and crossing over are going to be in prophase 1. That's where our legs are going to get stuck together, arms are going to get stuck together. Rather than untangling them, we're just going to cut them and glue them back together where they are. It's going to cause some variation. At the metaphase plate, again, remember in meiosis 1, there are paired homologous chromosomes, which we call tetrads, instead of individual replicated chromosomes. So we've got twice the DNA. And then at anaphase 1, the homologous chromosomes instead of the sister chromatids separate. So same thing, just more DNA. Continuing on our preview of differences, the first division separates pairs of chromosomes, not duplicate chromosomes. And then interkinesis is going to be present, so that's between meiosis 1 and meiosis 2. Meiosis is going to have 2 cell divisions, so it's going to be meiosis 1 and meiosis 2. All of our phase names will be anaphase 1, prophase 1, metaphase 1, and 2, all those types of things. I'm going to run through the steps really quick because they're essentially the same thing that happens during mitosis, but we've got twice as much DNA. So synapsis occurs as the chromosomes condense. Synapsis is where we're going to form our bivalents. We're going to form those tetrads. So our homologs are going to match up, basically. In prophase 1, again, we're going to see chiasmata, which is where we see crossing over. That's our longest phase of division, which should make sense because we've got the most DNA piled up in one place trying to sort it out. Metaphase 1, those bivalents and tetrads are going to align along the metaphase plate. Centromeres are going to point towards opposite poles. Anaphase, they're going to pull apart, but it's homologous pairs pulling apart, not individual chromosomes. Hopefully you remember this picture from Biology 1 that talks about independent assortment. If we get our two blue chromosomes over here on the left just by chance, we're going to end up with two sperm cells that look a lot like Dad, like Grandpa, and two that look a lot like Grandma. If the blue came from Dad's dad and Dad's mom. Over here in this possibility, we've got opposites. Just by chance, our blues are opposite one another. So this sperm cell is going to look a little bit like Grandpa, a little bit like Grandma. Same thing here. So we've got 4 different possibilities. They're all equally likely. In phase 1, the maternal and paternal chromosomes, now there's no difference between them. They just are chromosomes. Telophase, they're going to break up. It looks the same as it does during mitosis. So in interkinesis, no DNA synthesis occurs. This is between meiosis 1 and meiosis 2. May last for years, or the cell may immediately go into meiosis 2, depending on your gender. May appear similar to interphase of mitosis in terms of what's happening with the DNA and the chromatin and all those types of things. Meiosis 2 steps are the same: prophase, metaphase, anaphase, telophase. This looks a lot like the mitosis steps. However, the results are different. We have 4 cells produced that each have half the number of chromosomes. These cells are not exact copies of somatic cells. They're gametes. And then our genetic variation has increased drastically. That picture kind of summarizes all that. There's a table that summarizes all that. It comes straight out of your textbook, so I'm not spending a lot of time talking through those. 3 big sources of genetic variation in meiosis: independent assortment, we talked through. Random fertilization and crossing over, we'll talk through here in a few. So in humans, let's apply some of our math to the things to that picture I showed. We've got 23 pairs of chromosomes. So a chance to inherit 1 chromosome is 1 out of 2. You got a 50-50 shot. So think, I'm making the sperm cells for my children. My son has a 50-50 shot of getting my chromosome 1 from my dad, and a 50-50 shot of getting my chromosome 1 from my mom. That's what we're seeing here. Chromosome 1, chromosome 2. What independent assortment says is that these things are not linked. It's equally likely that my son gets my dad's chromosome 1 and my dad's chromosome 2. And it's equally likely that he gets my dad's 1 and my mom's 2. If we apply the math to that, there are this many possible combinations just from one person. So I can make that many different 8,388,608 different versions of sperm cells. My wife can make that many different versions of egg cells. Random fertilization, I really like this picture because it shows that fertilization is truly random. It's not a race. It's not the first sperm that reaches the egg. It's the first cell that's chemically compatible with the egg that gets in. Once that happens, all the doors lock, if things happen properly, and no other sperm cells get in. There are many stories of healthy couples that just can't have babies, and then they divorce or they break up or whatever, and they go their separate ways, and they each have children with other people that they are chemically compatible with. Tom Cruise and Nicole Kidman is an example of this. They both struggled for years to have kids. Then they split up, and it wasn't a very long time later before Tom had a baby and Nicole had kids. So, independent assortment, random fertilization. We'll pick up with crossing over in part 3.